and welcome to this afternoon's session on Resurrecting the Impossible, the Science and Ethics of De-Extinction, which has been much in the news this past year. Advances in gene editing and cellular engineering have transformed this once kind of impossible dream, as the title suggests, into a tangible scientific pursuit with implications for biodiversity, for reproductive science, for ecosystems resilience. And a lot of the running, most of the running, has been made by one company, by Colossal Biosciences, which just in this year alone have announced I don't know what you would call it, the rediscovery, the reinvention of woolly mice, of dire wolves, and most recently the resurrection of the dodo, so you can no longer say as dead as the dodo, or maybe not soon, and most recently the moa. And the company's also broken ground in commercializing this technology with more than $500 million in investment and a valuation which stands somewhere north of $10 billion, probably more than that by now. But there's still a lot that many of us don't understand about what's actually possible in this space, what remains beyond reach and might forever remain beyond reach, and how we might navigate these challenges, the social, ethical challenges of this technology as they emerge and develop. Fortunately, we have Matt James, who is the Chief Animal Officer of Colossal Biosciences here, to help fill us in on what we can do, what we can't do, what we should do. Matt.
Excellent. Thank you so much. I'm really excited to be here. I really appreciate your time, allowing me to talk to you about the exciting science of de-extinction. My name is Matt James. I'm the Chief Animal Officer at Colossal Biosciences, and you rightfully should ask yourself, what the hell is a Chief Animal Officer? If you are one of those people asking that question, I have the great privilege of working at Colossal Biosciences, the world's first de-extinction company, focusing on creating genetic engineering technologies to restore species once lost to science. As the Chief Animal Officer, I get to work with animals. I pet animals. I play with them. I'm responsible for the animal care, management, welfare of all the animals that we study today, the animals that we recreate for tomorrow, as well as I get to manage exciting fields of science like embryology and assisted reproduction. So, Colossal launched about 4 years ago with this idea that we're facing this massive biodiversity crisis in our world. And biodiversity loss is more than just the loss of intrinsically valuable nature. It's also a direct threat to humanity, the stability at which we live within this earth. But our food sovereignty, our water sovereignty is really dependent on having a well-balanced natural ecosystem. So understanding that the world might lose up to 50% of our biodiversity in the next 25 years, Colossal launched with this ambitious goal of being able to create technologies that allow us to restore species that have been lost to extinction. So we launched with this mission that was to make extinction a thing of the past, which is a really cute way of saying we can bring species back from extinction, right? But what's often lost, and what isn't talked about enough, is that to make extinction in the past is actually a two-fronted war. We can both reverse extinction, but we can also prevent extinction. The technologies that we create along the path to de-extinction, along the path to a direwolf, to a dodo, to a woolly mammoth, have direct application to biodiversity that are on the brink of extinction today. So we think about de-extinction, which is sort of a novel idea, but everybody, I think, has its own definition. The way we think about de-extinction is this idea that we can de-extinct core genes. We can focus on what were the genes that existed within the lost species that made it that species versus a close living relative of today. Can we compare and contrast their genomes, understand where that species is the same as the lost species and where it's different? Where it's different, we identify core genes, and we bring those genotypes, which result in specific phenotypes or physical traits, that perform function within a larger ecosystem. Our goal is to create functional replicates of these lost species so that when returned to an ecosystem, they perform very specific functions that help lift biodiversity, create stability within degraded ecosystems. Additionally, understanding that we are not engineering the past, we are engineering for the future, we focus on engineering enhancements and adaptive qualities into species that would not thrive in today's habitat, that have pressures of extinction present on today's habitat. So de-extinction is this idea that we have this end-to-end system, this pipeline of technologies, right? That starts with the really exciting field of ancient DNA. Every kid dreamt of digging in the dirt, discovering old specimens, and by doing that sort of paleontology work, our scientists can go and extract DNA, highly degraded, really poorly preserved DNA in these very ancient samples. We can create these really amazing genomes from this really poor DNA thanks to the modern technology of advanced genomic sequencing. We then compare and contrast those genomes to their closest living relative. In the case of a woolly mammoth, that would be an Asian elephant. In the case of a dire wolf, that's a gray wolf. We then identify which of those genes are most important, would be most impactful to creating the functional replicate of that lost species. And then we make those edits in living cell lines. Those cell lines are then produced into embryos, and those embryos are then moved into live surrogates, or maybe one day moved into biomedical devices that we call exodev, exogenous development devices, so that you can fully gestate an embryo outside of a mother's womb in a biomedical device. So quickly, this has garnered a little bit of attention. It's also helped us a lot on the fundraising side. In 4 years, actually this slide is a little out of date. I should have updated it for this. We have now raised more than $550 million in 4 years with a valuation of more than $10.2 billion. We started as a small team of 15, and there's now north of 180 of us. So it's been a rapidly developing, growing company. It's an exciting field of science. It's raised a lot of attention, which has helped us raise a lot of money. So quickly, we'll run through some of the projects that we are working on. If you haven't heard before, this year in April, we announced the return of these two guys here. This is Romulus and Remus, the world's first dire wolves. In 12,500 years, these guys were born via genetically modified gray wolf embryos that had the key dire wolf phenotypes and gestated in a domestic dog. I had the great privilege of being in the operating room when those first two fellas came out, and it was a life-changing moment. But what was really life-changing, I think, was the attention that followed. I spent 15 years of my life prior to joining Colossal working in nonprofit conservation and drumming up attention and awareness for really important conservation projects. And the return of Romulus and Remus resulted in more than 205 billion media impressions, 12.4 million social media engagements. Right? We had more than 50,000 unique journal articles written about it. So I think this is one of the sort of secondary, tertiary benefits that comes from these exciting projects, is that then we get to create conservation and science, and we're getting kids to use words like extinction, right? Instead of paying attention to the Kardashians or TikTok or whatever it might be. We're also working on maybe the biggest icon of extinction, the dodo, right? Native to Mauritius, it went extinct more than 400 years ago. This is an amazing project because the island of Mauritius is a biodiversity hotspot in which only 2% of its native habitat remains today. What's amazing is just by announcing this dodo project, by engaging with the Mauritian government, by engaging with the Mauritian people, we already have private landowners that want to turn their sugar plantations into dodo habitat. We have government that's interested in restoring habitat, removing invasive species to make room for the dodo one day. So this is another one of those benefits that begins to turn this engine to support habitat restoration, which is otherwise not an all too sexy project. The thylacine, or the Tasmanian tiger, is a famous extinction that occurred in 1936 when the Australian government exterminated the entire species because they thought they were a threat to their livestock industry. We're now just years away from welcoming the thylacine of the Tasmanian tiger back into the world. The island of Tasmania and the Tasmanian people are at the same time putting up fences, excluding invasive species, removing predators from their landscape in preparation for the thylacine. And then our iconic project, the one that we launched on, was this idea of the woolly mammoth, and it's the reason we're called Colossal. We're looking for big, audacious goals, really big, truly hairy goals. And I think the woolly mammoth is an amazing project because my background in conservation was working with elephants. What we've already done for elephant conservation, just in the pursuit of the woolly mammoth, includes creating vaccines against the deadliest disease that impacts elephants, protecting elephants in Samburu National Park, starting an orphanage in Botswana to help orphaned elephants return to the wild. And finally, we just announced this year that we're working with the Maori people of the South Island of New Zealand, the Ngāi to bring back an iconic species to New Zealand, the South Island giant moa, which, if you're not familiar, is a 14-foot tall flightless bird. But you hopefully are asking yourselves, why the hell would you do this? Right? Besides all the topics that I just mentioned, besides those secondary and tertiary benefits, one of the things we know is that rewilding is a very important conservation initiative that's happening. Actually, Europe and the UK are really leading the charge here. And they've shown that by restoring key species to degraded ecosystems, we've created these massive improvements in biodiversity, carbon sequestration, nitrogen cycles, natural-based solutions that help aid in our fight against global climate change. These species have that same impact. The other side is sort of what I alluded to earlier. The world needs de-extinction technologies. We live in a world today where we will lose 50% of our biodiversity in the next 25 years. Conventional conservation is amazing. It's where I put my blood, sweat, and tears for my first 15 years of my career. However, conventional conservation cannot keep up with the rate, the scale of biodiversity loss that we see today. Right? We're losing natural ecosystems. We're losing things to genetic bottlenecks, wildlife disease. We need to create technologies. We need to create support. We need to create underswell activity to support conventional conservation while accelerating and scaling it using modern technologies. So with that in mind, we launched the Colossal Foundation last year. This is a nonprofit foundation that I have the great privilege of being the executive director of, in addition to my role with Colossal, where we are literally where the rubber meets the road. We take technologies developed on the path towards de-extinction, and we apply them to critically endangered species today. So one of the things that got lost in that big direwolf announcement was that the American red wolf, the world's most endangered wolf, the only wolf endemic to the United States, which is also on the brink of extinction, now has this sort of genetic rescue component. We created a genetic bridge, a technological bridge that allows us to bring genes from coyote hybrid populations, take specific red wolf genetic diversity that no longer exists in their population, and restore it to them. So this little lady here, this is Neka Keita. She's the world's first cloned red wolf. We got 50 billion media impressions around dire wolves in that first week. In the first week, we also had 8 billion media impressions on words like red wolf. So really, really exciting. Similarly, we're working with the world's most endangered rhino, the northern white rhino. If you're not familiar, there's only 2 of these animals left in the world. They're a mother-daughter pair. They're functionally extinct. They cannot have offspring. So we've partnered with amazing people like Thomas Hildebrand at IZW in Berlin to use assisted reproductive technologies, to use the southern white rhino as a surrogate to make more northern white rhinos. However, if you only have 2 animals, if you only have 12 cell lines, you're going to be very limited based on the genetic diversity. So we're working in museum samples. We go find people that had poached these animals 100 years ago, go work in museums, the natural history museums, extract DNA, understand what was the genetic diversity prior to the bottleneck, identify key alleles within that population, and restore them into cell lines today. And if you're not familiar, the largest threat to biodiversity in the world is a little fungus called chytrid. It's now present on every continent where amphibians live. It's already driven 95 amphibian species to extinction. And we are looking at another 500 species that are threatened by this horrible disease. Using genetic engineering technology, we've identified key antibodies that exist, strangely enough, within alpaca. We can take those transgenes, put them into endangered populations of frogs, and suddenly these frogs that were surely going to go extinct are now resistant to the largest threat to their life. And then working in Australia with one of the world's most endangered marsupials, this is the northern quoll. It's very cute, but it's a tiny, very hungry carnivorous marsupial. It predates on that famous cane toad, which was an invasive species introduced to Australia to help eat all the cane beetles. Well, they didn't eat cane beetles, they ate everything else. And unfortunately, when these quolls predate on that cane toad, they ingest a toxin called bufotoxin, and they die. So we're quickly seeing this population crash. By creating one single base pair change, we've identified a gene that would make them 500 times more resistant to that bufotoxin. This work's already occurring in the lab. We're already engaged with the Australian government to identify experimental regions where these animals could be released to help save this population from sure extinction. So what we've done, which I'm really proud of, is sort of taken all these amazing technologies, this exciting idea of de-extinction, and we've created this disruptive framework for conservation. Using all the technologies that we need to bring a woolly mammoth back from extinction, we can independently deploy those in this chain of events to go out, identify the world's most endangered species, identify species that we thought were lost to science. Once we find them, we can biobank them. We can sequence their genomes. We can create these reference quality genomes that then every scientist in the world that wants to access that data can use. So that becomes a really powerful tool, and then we need to look at things like engineering vaccines or gene engineering tools to create resistance for critically endangered species to things like bufotoxin or chytrid. And then finally, species that are on the recovery sort of lift, they need a genetic boost. In addition to demographic recovery, we need to bring genetic diversity into these populations. This data set, these tools allow us to do exactly that, just like what we're doing with red wolves and northern white rhinos. And then finally, with all these media impressions, with all this fundraising, people say, shouldn't you be spending that on conservation? Yes, absolutely, and that's what we're doing. We use that to then fund habitat restoration, rewilding efforts, begin to support conventional conservation. So all that to say, I often get that remark of, wouldn't it be great if you used that $500 million to support conservation? And I say we are. De-extinction is conservation. Conservation needs a tool like de-extinction to succeed. The two are intrinsically linked. As we support conservation and build new rewilding tools using amazing tools of de-extinction, we then create this value chain that supports it from the bottom up. So thank you very much. I really appreciate your time, and I look forward to what should be an interesting set of questions.
So normally, this is the point at which the moderator indulges his vanity by asking questions of the speaker. But I think we've probably already got a lot of questions in the room. Put your hand up if you have a question. OK, so let's just go straight to that, please. The gentleman here was the first one I saw. On the aisle just here, please. If you could stand up, please. Can you stand up to ask your question, please? And maybe briefly introduce yourself very quickly. Thank you.
Yes, my name is Gabor Farkas. I'm a mediator. My question is, how far back do you expect to go? And when will you think of bringing back Neanderthals?
Well, luckily, I'm the chief animal officer, so I get to stay away from Neanderthals. So right now, we can basically go as far back as ancient DNA allows us. The oldest ancient DNA sample that we've been able to sequence is about 1.2 million years old. So that would mean anything that went extinct in the last 1.2 million years, assuming proper preservation of that sample, could be sequenced and one day restored. But technology advances every day. So you could eventually maybe get more beyond that.
Lady here, please.
Hi. Yes.
Oh,
Hi.
okay.
Carmen Saldana from the University of Barcelona. I have a question. You've touched upon it in the last slides, but I was wondering if you're doing the same effort of rewilding or the genetic technology that you're developing for the habitat itself, or you're just doing that in conservation? Because, I mean, the habitat changes through many years also with the de-extinction.
Yeah, exactly. So we work in networks of NGOs and government partnerships in order to prepare habitats for these animals. You won't often see boots on the ground from somebody in a Colossal sure doing that work. That's often done through partnerships. So, for example, in Mauritius, our habitat restoration work for the dodo is being performed by the Mauritian Wildlife Foundation, a partnership that we've developed, we fund, and we bring our technologies to. For one project that we're working on with them is the genetic rescue of the Mauritian pink pigeon. So the dodo is leading to the genetic rescue of the most endangered pigeon on the island of Mauritius, and Mauritius Wildlife Foundation is restoring habitat, ensuring that all the drivers of extinction there are being removed so that the pink pigeon can thrive, and one day maybe the dodo.
OK. Just here. Oh, sorry. Beg your pardon.
Hi, Audrey Yimmer from the Open Quantum Institute. My question is more on the adverse effect. Did you ever have to balance the impact of the reintroduction of a species and how it could impact perhaps negatively some other species?
Yeah, absolutely. I think a big piece of what we do is we develop what we call these conservation assessment and risk evaluation workshops. So we work with an amazing group called Advanced Conservation Strategies led by Dr. Josh Donlan, and we sit down and work in networks of experts in that region with those taxa to identify what are all the upside consequences and what are all the negative consequences. How do we mitigate the negative and how do we accelerate the upside? So those things do not happen quickly. They don't happen in a bubble. So every one of the species you've heard about today, there's already a panel of 3 or 4 experts that are developing those guidelines for us today. In addition, we work with the IUCN Species Survival Commission in order to drive an update to the de-extinction guidelines that can show exactly how that process should occur.
Hi.
Could you stand to ask your questions, please? Sorry, it's for the online audience. Apparently, this makes it easier.
No problem. My name is Lauriane Trimoula. I work for the Gallifrey Foundation. It's an ocean conservation organization. So still on the adverse effect, is we're very much into shark protection, for instance. So I'm wondering if this solution is not sort of authorizing overfishing more and more, because who cares about the shark? There will be a solution to de-extinct them again or recreate them again. So I have my doubts, to be honest. And given the fact that we exterminate sharks 100 million per year, that's 270,000 per day, I have very mixed feelings about this, to be honest.
I think that's totally fair, and it's one of the things we consider is that moral hazard of if you have a technology that allows you to restore a species from extinction, why should anybody care about extinction? Why should you protect nature if you could just flip a switch and bring it back? I think number one, it's because that switch is really hard to flip. But number two, my experience, at least for almost 20 years working in this space, was that people didn't care enough today, right? I think we need to find ways to drive this next generation to care more, to be more inspired, to get away from some of the doom and gloom that 24-hour news cycles can provide, this sort of sense of helplessness, create awe and inspiration, drive a connection to nature. But to your point, we have to also be really responsible with the way that we talk about the technology. It is not simply bringing species back from extinction, and often we can't restore 100% replicate of that animal. So once that animal is lost, it's important the public knows that that animal is truly lost. We can bring back functions, but we can't bring back individuals. We can't bring back exact replicates.
May I? Thank you very much. I'm Supriya Bezbarua. I'm from WHO, but this is personal. I'm also a molecular biologist originally before I moved. So this is absolutely fascinating, and clearly you've made your case for how important it is to have the de-extinction technology, because half of all species will be extinct by 2050. My question is one of speed and balance, and it's kind of related to what she said, but not exactly. Here you are, you have species going extinct, and then here, on the other hand, here you are sort of de-extincting them on the other hand. Can you catch up? I mean, extinction is caused by various complex issues, right? And de-extinction is needed, but is that enough? So are you also looking at the same time, using technologies to prevent extinction? How do you manage the speed of de-extinction, so to say, to prevent...
Absolutely. I mean, it's a big piece of my job is to understand how we can slow the rate of extinction, how we can apply our technologies in a meaningful way to sort of slow extinction to a point that where maybe it's more sustainable as we catch up with de-extinction. I think it's important to note that at no point does Colossal want to give the impression that this is a silver bullet to our biodiversity crisis. This is a silver bullet to extinction. These things have to happen hand in hand with conventional conservation. The foundation that I mentioned, we've raised more than $100 million to support conventional conservation while applying our technologies. One of the things you'll see out in the world, the Paulson Institute did an amazing study about 5-6 years ago that showed globally we need to invest about $900 billion a year to stop extinction in its tracks. Globally, right now, we average about $180 billion spend on conservation. So we have this $720 billion delta. We need to invest in technologies that makes conservation, biodiversity protection more efficient. We need to bring that $900 billion number down while also bringing more money into the equation and try to close that chasm of investment. That's not going to happen without innovative technologies, and it won't happen without new players coming into the conservation space. We've raised $550 million. That's $550 million that would have never existed for the conservation space. My CEO likes to say, all we did was we prevented another shitty software company. Right? We took money from Silicon Valley that otherwise would have gone into a really bad product, and we created this company. And now that's money that's novel to the conservation space.
Hi, my name is Maxim, and I'm a high school student here on behalf of the Villars Institute. And my question was, to what extent can de-extinct species really replicate their extinct ancestors' behavior and physiology, given like the differences in the surrogate species and also the differences in the current modern ecosystems that they have to adapt to?
That's a fantastic question. I love that. I think your question is sort of alluding to a point that you've already probably concluded, is that it is impossible to replicate the exact physiology and behavior of an extinct species. What we're focused on is creating extinct functions for tomorrow's ecosystem. How can we find animals that will thrive today and tomorrow, not yesterday? So we focus on working with amazing groups like that elephant orphanage in Botswana I mentioned. How do we take a 3-year-old orphaned elephant, put them into a peer group, and get them ready to be their own cohort of individuals, create a herd, a synthetic herd, and put them back in the wild? That type of opportunity allows us to protect species today, but also learn a lot along the way about how you should be rewilding animals that don't have a mother. So we find projects like that, and then we also focus on what are the ecosystems of tomorrow that they need to thrive, and that's where we're focused.
So Robin Littlebatch, that was going to be one of my questions. We discussed it a bit yesterday anyway, but the other one is, how do you choose which alleles to modify? So for the dire wolf example, was it 14 or something like that?
Yeah, it's 20 edits, 15 genes.
Yeah, but there are many, hundreds of thousands of genetic variants between a grey wolf and a dire wolf.
Yeah.
And how do you know which ones are going to be more relevant to giving a real dire wolf in terms of its how it interacts with its environment, other species in its environment, prey, its behavior, etc., etc., etc.
So with the limited knowledge we have of dire wolves, right, up until the point that Colossal invested in this, we had very little genomic data. All we had was basically skeletal and fossil remains to make interpretations of behavior. We then sequenced the DNA. What we could tell from the DNA by comparing them to closely living relatives, we then extrapolated that information using computational biology SOP4 platform, AI-powered tool that allows us to sort of make educated guesses about what each of these genes do. How do you get this sort of physical replicate of it? If you can replace physical traits, you can make assumptions about ecosystem functions, and then it takes, obviously, generations of studying these animals in these habitats. These animals live on a 2,000-acre habitat where they're studied every day, and then that helps inform the process of de-extinction. There is no way to look at a genetic sequence today and say that's responsible for a hunting instinct, right? We have to go and see how these things play out in the real world as well.
How are they?
Well, I mean, they're still quite young. They just turned 1 year, which was a really exciting milestone, but they are behaving sort of like you would expect a large predator. They just actually, the really most exciting story that just came out about 3 weeks ago is my team called me and they showed me a picture of Romulus who was covered in blood. Right? And he went and he got his first kill. And so he's learning how to hunt. He's learning how to do these things. And it was, for a bunch of animal people to celebrate this guy killing another animal is weird, but it was also a great sign that these guys are really developing the instincts and the tools that we thought that they would need in order to survive in this semi-wild habitat.
We're running up against time, so I want to take 3 questions really quickly, please. Here, at the back, and then at the back. Sorry, you're going to have to run around, but here, there, please. Quickly, please.
Yeah, David Kong from IT. Can you just comment a little bit about the social dimensions? So, how can you ensure that communities and the people on the ground either want these species, and just challenges and innovations on the social side of how you've been able to really bring these animals out into the wild?
So when we get the tools in place, we get the resources in place to say, okay, the de-extinction of this species is possible. Our next step isn't science. Our next step is actually social science. We go and plug plug in with local communities. So the MOA is a great example. We tied in with the Naya Tahu Research Center, and they're leading social science and engagement, also governmental engagement and regulation. So in Mauritius, we have a Mauritius Dodo Advisory Council. In Tasmania, we have the Tasmanian Tiger Advisory Council. All these are really meant to bring local stakeholders to the group, have diverse representation from indigenous peoples to industry to academia to tourism, and we bring those people together and help sort of hand the keys of the project over to them locally while we work in Texas on the science side.
And back there, please.
Yes, thanks. Very interesting. One question about intellectual property and who owns and how do you manage this intellectual property of all these species that you make live again?
Yeah, it's a really good question. It's a hard one to navigate, right? Who owns genetic data? Who owns all the information you can extrapolate from these animals? And that's part of what we work on the international regulatory front when we're working at CBD or CITES, working with things like benefit sharing and Nagoya protocols. That's what we're focused on: is how can we build a benefit? How can we share in the knowledge? Just sequencing DNA and publishing it publicly doesn't really create equitable access to that data. So one of the things we do is we focus on capacity building. So we have a conservation project that we're about to launch in Indonesia with Javan rhinos. So first and foremost, what we're doing is building sequencing and biobanking labs in Indonesia so that the Indonesians can help lead that project and have that direct benefit.
Hello, I'm Alex Antonelli. I work with GESDA on the Eco-Augmentation Initiative. My question to you is related to regulatory frameworks. What would you like to see for your company and others to play an ethical game and really be able to advance this in an inclusive and impactful way?
I think, if you're familiar with the IUCN, they just had the World Conservation Congress, and at the Congress there were two propositions that were up to vote. One of them was a flat ban on using synthetic biology as a tool for conservation, genetically modified animals on the wild landscapes. That was luckily shot down because I think it's a short-sighted ban. I think what I'd like to see on the regulatory front is the creation of sandboxes, these international places where we can work together to experiment on innovative technologies, understanding that every day we wait, every day we talk about regulation and we don't act, we're falling further behind. So we need to create these opportunities where we can work together in a safe place to develop tools, innovate, while also developing the regulatory pathways in parallel. If we put these in sequence, we'll lose. We have to figure out how to invest in research today while creating regulatory pathways to success.
Okay, I think there's probably lots more questions in the room, but I'm afraid that is somewhat beyond time. So thank you very much for coming, both here and those of you watching online. There we go. Please do complete the survey, the 1-minute survey about what you thought about this session. And if you'd like to contribute to the conversation online, you can do that under the hashtag GESDA Summit. It only remains for me to say thank you very much to our speaker, Matt James. Please run with the